Now, the electron transport chain is not controlled hormonally. Rather, it's controlled by the energy level within the cell itself. When ATP builds up within the cell, which is a sign of high energy, the electron transport chain actually slows down. And when ADP builds up, which is a sign of low energy, the electron transport chain speeds up. Now, it turns out that drugs and chemicals can effectively break oxidative phosphorylation in two ways, either uncoupling or inhibition. Let's start with uncoupling. Normally, the electron transport chain is coupled with ATP synthesis, meaning that they happen together. Uncoupling agents break that link, and they do this by inserting their own proton channels, called ionophores, into the inner mitochondrial membrane, or by simply carrying the protons back into the mitochondrial matrix, thereby allowing them to bypass the F0 subunit of the ATP synthase enzyme. Uncoupling agents dissipate the proton gradient created by the complexes. And because the protons enter back into the mitochondrial matrix with the uncoupling agent instead of through the F0 component of ATP synthase, F1 isn't able to phosphorylate ADP to make ATP. Now, it's important to note that the electrons are still flowing from complex to complex all the way to the final electron recipient, oxygen. So that means red blood cells keep delivering oxygen to the tissues so that it can be the electron recipient. Also, as the cell's ADP levels rise, the body tries to increase its metabolic rate to make more electron donors like NADH and FADH2. But this electron flow is essentially useless, because no matter how large of a proton gradient there is, the protons just go back with the uncoupling agent instead of through F0. And because some of that electron's energy is not used up in moving protons across the inner mitochondrial membrane, more of it is available to turn into heat energy. There are some endogenous uncoupling agents like thermogenin, which is a protein found in the brown adipose tissue of babies, and is used to generate heat. Funnily enough, thermogenin is also found in hibernating animals. So yeah, we're kind of like polar bears when we're babies. Uncoupling is kind of like a car with a hot, hard-working engine, but despite that, the car isn't moving. Another example of a medication that can be an uncoupling agent in high doses is aspirin. The uncoupling can lead to a really high metabolic rate, which can lead to a metabolic acidosis. And if ATP levels start to fall too low to allow the respiratory centers and respiratory muscles to work, then it can lead to a respiratory acidosis as well. A combined metabolic and respiratory acidosis quickly becomes life-threatening. Alright, the other way that oxidative phosphorylation can go awry is by inhibition, meaning that some chemicals and drugs inhibit components of the electron transport chain. Unlike uncoupling, inhibition stops the flow of electrons through the electron transport chain, and that leads to a decrease in ATP synthesis. Because electron flow is put to a stop, electron donors like NADH and FADH2 build up, so the body doesn't feel the need to make more, and the metabolic rate actually falls. Inhibition is like turning off the car engine, which of course means that the car still won't move. And not surprisingly, poisons that work by inhibiting the electron transport chain can lead to death pretty quickly. Examples of inhibiting agents include carbon monoxide and cyanide, both of which inhibit complex 4 of the electron transport chain. Barbiturates, which are GABA agonists that can be effective for seizure disorders, inhibit complex 1 at high doses. Oligomycin, which is an antibiotic too toxic for human use, inhibits the F0 component of ATP synthase. Statins, which are a class of lipid-lowering medications, don't inhibit the electron transport chain, but they can decrease the synthesis of coenzyme Q. And that reduction in coenzyme Q can result in decreased ATP production and lead to muscle pains and cramps, and rarely can even cause rhabdomyolysis, which is when there's severe muscle breakdown which results in kidney failure from the myoglobin released.